Cellular respiration. Introduction by Navi and Darlene. What is cellular respiration? It is a way for cells to get energy from organic molecules, and the main goal is to make ATP. The most commonly used organic molecule is glucose. The major stages of cellular respiration include glycolysis, the citric acid cycle, and oxidative phosphorylation, which is broken down into two steps, the electron transport chain and ATP synthase. Glycolysis takes place in the cytosol. The citric acid cycle and oxidative phosphorylation takes place in the mitochondria, and about 32 to 38 ATP are produced. Glycolysis is the first stage in cellular respiration. It is 3 phases long and with a total of 10 steps. This creates a net total of 2 ATP and takes place in the cytosol. The energy investment phase. This is the first step, a major step, and in glycolysis, and 2 ATP are hydrolyzed to create fructose 1,6-phosphate. Then the cleavage step occurs, where the 6-carbon molecule is broken down into 2 3-carbon molecules of glyceraldehyde 3-phosphate. The energy liberation step occurs, and 2 glyceraldehyde 3-phosphate molecules are now broken down into 2 pyruvate molecules. The citric acid cycle is the next major step in cellular respiration. It takes place in the mitochondria, and pyruvate is converted into acetyl-CoA and 2 NADH are produced. In the citric acid cycle, 2 ATP, 6 NADH, and 2 FADH are produced. This is the citric acid cycle. As you can see, the acetyl-CoA combines with the oxaloacetate using the enzyme citrate synthase to become citrate, which then uses an enzyme to become isocitrate, which uses another enzyme called citrate dehydrogenase to become alpha-ketoglutarate, and 1 NADH is produced. Then, the alpha-ketoglutarate using alpha-ketoglutarate dehydrogenase becomes succinyl-CoA, and another NADH is produced. And the succinyl-CoA becomes succinate, and using another enzyme, and the succinate becomes fumarate using succinate dehydrogenase, which then, with the addition of water, becomes malate. And malate using malate dehydrogenase once again becomes oxaloacetate, which then combines with acetyl-CoA to make do the cycle again. Okay, the electron transport chain. NADH reduces protein complex 1, or NADH dehydrogenase, and the electron travels to ubiquinone and goes to cytochrome dehydrogenase, and the cytochrome C, and the cytochrome oxidase, and to the final electron acceptor oxygen. FADH can also be used in the electron transport chain, but it gets oxidized through a protein complex farther down the electron transport chain, so less energy is captured. Okay, ATP synthase. The electron transport chain creates an H+ gradient in the inner mitochondrial membrane. The H+ cannot diffuse through the membrane to get back to the mitochondria, and active transport is required by the use of the protein ATP synthase. While moving the H+ to the mitochondria, ATP synthase combines with the ADP with the phosphate to make ATP, and about 32 ATP are made from this stage. Okay. Enzymes are used all throughout cellular respiration. One of them are a group of enzymes called kinases, and those are enzymes that transfer phosphates. An example in cellular respiration and the use of kinases are during glycolysis when high-energy phosphates are added to the glucose molecule. Then there are dehydrogenases, and those are enzymes that oxidize their substrates. An example of dehydrogenases are in the electron transport chain, the protein complexes, some of them are dehydrogenases. Then Also, enzyme pathways can be controlled with excess or depletion of a certain molecule in the cell. Phosphofructokinase is one of the most highly regulated enzymes in the cell, and stops when there are high levels of ATP, but then starts when there are high levels of ADP. This is because what phosphofructokinase does is prevent the third step, catalyzes the third step in glycolysis, which breaks down the glucose. And if there are high levels of ATP, then the glucose doesn't need to go through cellular respiration, so that way the ATP can stop it from happening, and the glucose can be converted into fat and stored for a later time. However, when there are high levels of ADP and a lot of ATP needs to be produced, the phosphofructokinase starts again and cellular respiration can take place in the cell. Many different macromolecules, glucose is the most commonly used macromolecule for cellular respiration, but other macromolecules can be used, like proteins or lipids. Lipids can be used as glycerol or fatty acids. The glycerol can be converted into glyceraldehyde 3-phosphate and then used in the cellular respiration. And fatty acids can come in as becoming acetyl-CoA and then used in the rest of cellular respiration. Proteins become amino acids and then can either come in as pyruvic acid, as acetyl-CoA, or in the Krebs cycle to finish up cellular respiration.